Okay, good morning, everyone. Welcome to day 3 of A of A 2025. The end is near, or more precisely, the ends are close. So our first speaker is Torin Greenwood from North Dakota State University, and he's going to explain to us that the ends of a branch structure are necessarily close.
Thank you very much. Thanks to the organizers for organizing everything and for allowing me to speak, and thank you all for listening. So I'm going to tell you today about joint work with Christine Heisch that was motivated by some recent work in RNA folding. And so the first thing I'm going to do is remind you a little bit about a very simplified version of what RNA does. And so there's this central dogma of molecular biology, which says that DNA strands within a cell create RNA, which then creates proteins. And so it was, well, there are two things. One of them is that DNA is highly structured in that it forms this double helix, and it always forms a double helix, whereas RNA and protein are a little bit more complicated because they fold up into three-dimensional jumbles. Protein folding is an area of research on its own that people are very active in. RNA folding, also an area, and maybe provides an intermediary in the sense that it's not quite as complicated as protein, but still much more complicated than DNA is. And then the other thing is that RNA really has become an area of research all on its own because you can't exactly tell from DNA what's going to happen with this whole process. Because, for example, when DNA is read off into RNA strands, there's often some processing that goes on after it's read off before it becomes the strand that's actually going to do something biologically. And so one reason why that might happen is, for example, because a virus might embed itself into DNA, and then that's still read off into the RNA, and then the virus portion is cut out. And so for that reason, we really do want to study RNA directly. Okay, and so then we could talk about how are we going to study RNA, and one way is to look at the structures that it folds into. And the reason why you might want to do that is because if you have a database of sort of known RNA sequences and you know what their structures are, then if you find another RNA sequence and it has a structure that looks similar, then you can probably guess it does the same thing. And so on the right, there's this picture of a 3D representation of tRNA. And you could use X-ray crystallography to figure out, that's sort of the gold standard for figuring out what the structure is, but that's expensive and slow. And so we would rather come up with a method that just uses the one-dimensional strand of nucleotides on the left and does some sort of prediction algorithm to guess what the 3D structure is going to be on the right. But maybe guessing a 3D structure is a little bit ambitious. And so instead, people often look at this secondary structure in the middle. And so it's a flattened version of the 3D structure where the rules are that every nucleotide can pair to at most one other nucleotide. And then also the nucleotide pairings can't cross over each other. So if I look at that line right there, I couldn't have this G like pair with that C and then this C pair with that G because it would form an X in terms of the pairings. And so the important part here is that secondary structures have been known to still encode enough biologically meaningful information to like determine the function of various structures. Okay, so with that in mind, one sort of big question would be if you just have sort of a random sequence of A's, C's, G's, and U's just as a word, and maybe you even have like a guess of a structure for what those could form into, then how could you tell whether that's biologically realistic? And so what is it about biological sequences and biological structures that are maybe different from random sequences and random structures? And so of course, there's a ton of ways that you could approach this. One thing you could do is just look at the sequence of nucleotides of A's, C's, G's, and U's and try and compare like nucleotide frequency to actual strands or maybe tuple frequency. And so there's a reason why you might want to consider tuple frequency, which is if I look at like groupings of 3 nucleotides in a row, then those correspond to codons, and that's actually how like amino acids are encoded in RNA. And so maybe I want to look at 3 at a time and see what the frequency is there. And that sort of work, has been certainly explored, well explored. And another option would be, maybe we have a structure that we can guess it folds into, like the secondary structure pictures I was showing you before. And maybe we can look at the different sort of biological features of those structures and see how likely it is that random structure has something versus true biological structure from the databases of RNA. So I'll show you a picture of some biological features that we might care about. And so this is taken from Christine's work with Svetlana Plazanovic. And so it's a picture of an RNA secondary structure, and just some of the features that come up in biology a lot are marked here. And so one of them, whenever we have like a consecutive run of base pairs up top, we'll just call that a helix. And so we might care about how many helices there are and what's the length of the helices. Another sort of thing would be a loop. And so at the end of any arm, if there are a bunch of unpaired nucleotides, then we call that a hairpin loop. Or if there's a spot where it's branching into more than two things, we call that a multi-branch loop. And then there's some other sorts of structural features you could look at as well. This is certainly not exhaustive. So those are the names for the start and end of the like one variable or one dimensional sequence. Okay, so I'm going to focus specifically on one type of feature because of this recent work from some biophysicists where they were looking at large RNA molecules, and they concluded that the ends of those large RNA molecules always ended up being close to each other once the sequence had folded. And so I'm going to show you a picture from their work, Yashi et al. So here's RNA secondary structure that has like 200 nucleotides in it on the left. And then on the right, I'm zooming into just the area that sort of captures the beginning and the end of the sequence. And so that's over here now. And again, the 3' and the 5' are just names for the start and the end of the sequence. And then maybe what we care about is what's the shortest path that you could take between those where you're allowed to jump over like base pairs. And so any two nucleotides that are paired together. And so if I follow that path right here, then I can see that the length is 16. And then another thing which we'll talk about today is that there's only one base pair in that path, which eventually we'll refer to as degree. And so the point here is that this was a relatively large sequence with 200 nucleotides, but the distance between the ends was only 16, and there was only one branch off of that path. Okay, and so the conclusion is that they found that this was true not only for this example, but also for databases of real RNA fragments. And then also they did some random analysis by taking a known RNA sequence, shuffling all the nucleotides in it so they're in new positions, and then finding the minimum free energy structure, which you can do with existing algorithms, and found that the ends were still close there. And so then the question for today is, like, how surprised should we be about the fact that these ends are close? And of course, the level of surprise should really depend on what we think the background distribution is for RNA structures. And if you're viewing RNA structures sort of as a three-dimensional jumble where you start with a strand, you go all over the place, and then you end wherever you end, then maybe it is pretty surprising that the ends are pretty close to each other in the end, because you'd expect maybe they'd be on the opposite ends of the structure or whatever. But we're going to use some sort of combinatorial models to look at this in a different lens. Okay, so I'm going to tell you about a couple ways that we might encode things here. So on the left, I've just drawn a schematic of an RNA secondary structure. And one way we could encode it is in a rooted plane tree. And so the way that we would do that is, first of all, I have the ends of the sequence now at the bottom left and the bottom right. And that I'm now going to refer to as the exterior loop. And below the exterior loop, I'm just going to consider that there is a node right there. And then I'm going to look above that to all of the base pairs touching the exterior loop. So there's two right there. And those are going to become my first nodes in the tree. And then from this one, I can see that branches into two more groupings of pairs. And so those become additional nodes. And so if I sort of flip that upside down, then I get this rooted plane tree right over there. And so I can see that this is really a compression of information because, first of all, I'm not keeping the number of base pairs in a helix anymore. That's always compressed to 1. And then I'm completely ignoring the unpaired nucleotides. And so I get these rooted plane trees, which of course are also in bijection with dict paths. And the way that you do that is you just take the path around the rooted plane tree. And every time that you take a step away from the root, that corresponds to an up step in the dict path. And whenever you take a step towards the root, it corresponds to a down step. And dict paths, as a reminder, are paths that take steps up and down and always stay above the line y equals 0 and both end and start on that line. So that's one option for how we could encode these RNA secondary structures. And I should mention that this is an option that has been explored for other reasons in RNA. And so it's sort of an accepted combinatorial model for RNA secondary structures. Another option which would not lose information, it's a bijection, is to just read along the edge of this structure. And every time that I have unpaired nucleotide, I'll store that as a horizontal step. And then every time I have a paired nucleotide, it'll either be an up step or a down step, depending on whether it's the first or second nucleotide in that pairing. And so I could also equivalently store that as a dot bracket sequence where the dots are just the unpaired nucleotides and brackets are sets of paired nucleotides. Okay, so we have these representations, and now I want to trace through sort of what does the various features of the exterior loop correspond to for these representations. And so the first thing I might look at is just the number of base pairs in the external loop. Right? And so that's, in this case, it's just 2 because there's the one base pair right here and the other base pair right there. And if I push that through to the tree version, then not surprisingly, that corresponds to the degree of the root, which is a well-studied thing, but we will go through that just to see sort of the structure of the analysis for more complicated things. And if I push this forward into the level of paths, then both for the dict paths and Motzkin paths, it's just telling you about the number of mountains in those dict or Motzkin paths. And so now I'm going to be interested in, if I consider all dict paths or all Motzkin paths, then what's the distribution of the number of mountains in those paths? And then I can compare that to what happens for these RNA sequences. Okay, and so I'm going to need some sort of analysis pipeline in order to figure this out, and I'll use analytic combinatorics, of course. And so I'm going to first try and store things in a bivariate generating function, where in this case, d of zu will count the number of dict paths of length n, which have k mountains, where n is just the length of path and k is the number of mountains, and z tracks length and u tracks number of mountains. And then I'm going to look at these probabilities right here the way that you normally would. And so I'm going to look at the coefficient of z to the n, u to the k in d. And so that's the number of paths again of length n with k mountains, and divide that by the total number of paths of length n, which is given by this coefficient. And so then I want to look at what the probability distribution is as n goes to infinity. And so I want to look at this limiting distribution. And of course, eventually I want to justify that if I look at the limiting distribution, it's still realistic for the length of RNA. Okay, so the first thing I need to do is find the generating function, which I can do sort of with the standard dict path specification in this case. And so I can break down any dict path into 2 smaller dict paths according to its first return to the line y equals 0. And when you do that, you get these 2 legs right here, and then an arbitrary dict path here. And this has to be a dict path that's staying above that line, because if it went below, then the first return would have been earlier. And then I get an arbitrary dict path after that as well. And so I can just convert that into an equation. And so any dict path is either empty, that's where that 1 is coming from, or I can decompose it, in which case the 2 legs add length 1. I get 1 mountain from this first dict path right there. I can put the univariate generating function here because there are no additional mountains from that first blue path. And then I have an arbitrary dict path after that with as many mountains as there weren't. Okay, so this determines that generating function. And now I want to solve for it. Again, a relatively simple case, but we can solve for this and we get this fraction right here. And the important features about this fraction are just like the univariate case. If I'm doing singularity analysis, the part that matters is the stuff underneath the square root. And in this case, the stuff under the square root ended up being a function of z alone, because the u stuff ended up being a perfect square that factored out of the square root completely. And so that'll be a theme that runs through all of the examples that we look at, which makes the analysis quite convenient. And from Flagellate and Sedgwick's book, in chapter 9, we know that if this is the sort of scenario where the singularity is of a fixed exponential rate and it doesn't depend on the variable u, then we should be expecting a discrete limiting distribution. Okay, so now I want to figure out how am I going to actually get what the discrete limiting distribution is. And again, we could sort of do this through standard singularity analysis. And so this is really just packaging the standard techniques in a form that I can use for all the examples that we have here. But this is really saying if you have any quadratic generating function that's of this form where the stuff under the square root is one variable and the u stuff factors out, and then you need some sort of condition that says that the singularity of this part is what actually contributes to the asymptotics in the long run, so you're ruling out other things. Then with those conditions, the limiting distribution is really simple. It's just a ratio of g of u divided by g of 1 times a evaluated at the singularity of the univariate part. And then again, you plug in a u and a 1. And so the point is, once we have the generating function, if we can verify this fact about the univariate singularity, then we can just plug into this formula and we get the limiting probability generating function. Okay, so let's go back to the dict path example and see what happens when we plug it in here. So in this case, the u function right here, g of u, is just plain old u. And then a of zu is this full denominator right there. And then the singularity is at z equals 1/4. And so I'm just going to plug in those things into this fraction, and I end up with this probability generating function right there, which in this case is 1 plus a negative binomial distribution. And so negative binomial distribution just means that you are flipping a coin until you get 2 tails and counting how many times you had to do it. And the 1/2 right here means that the coin is fair. And so in this case, the mean is 3 of this distribution and the variance is 4. And so it's not particularly surprising from this viewpoint that the exterior loop degree should be small in an RNA secondary structure. One quick comment I wanted to make before moving on. Like I mentioned, the number of mountains in a dict path has been studied before. I can't see anything about combinatorial interpretation in the literature for why flipping coins has to do with counting mountains. If you have anything about that in the literature or on your own, then please do let me know. Okay, so one other aspect I wanted to, or I'd mentioned earlier, is that we do need to make sure that this convergence is sort of fast enough that it would actually apply for RNA secondary structures. Of course, we could use analytic combinatorics to come up with error rates, but we can also plot things for small values of n just to make sure that even for reasonably small RNA structures, this is still converging quickly. And so this is just a graph to show the black points are this limiting negative binomial distribution. And then the blue, red, and orange are just for dict paths of length 10, 20, and 40. And the point is that even for n equals 10, this is a reasonably good estimate of the limiting distribution. And so we should expect RNA to really closely follow this. Okay, so I just told you about degree, which is one measure of the exterior loop's complexity. But we could also look at the length between these endpoints, which would just be all of those nucleotides there. For the dict paths, that ends up just doubling what we had before. And so it's not very exciting compared to what we talked about. But for the Motzkin paths, there's the additional content of this is really counting all of the steps that are either on the horizontal axis or an up or down step that one end touches the horizontal axis. And so with the Motzkin paths, then we can still push through the same analysis pipeline and figure out what the distributions are. And so for Motzkin paths, the degree again tends towards the same negative binomial. And the length tends towards something that's a little bit grosser. And so it has this probability generating function, but you could identify easily from that, of course, anything about the distribution, but that the mean is 8 and the variance is 28. And so the example that we saw before, which had the length between the ends being 16, is not out of line then with this probability distribution. I want to mention that there's a lot of work already about sort of time spent on the line y equals 0 for various paths, although the parameters are slightly different, but a lot of work for different path families by Banderia and Wallner as well from 2018. Okay, so the conclusion so far is that for these combinatorial models of branching structures, maybe not surprising that the loop has low complexity. But you could argue that maybe I've simplified things too much. And so maybe I want a slightly more realistic model of RNA, and I still want to conclude from that that the exterior loop has low complexity. And so another existing model in the RNA literature is to use a context-free grammar to generate RNA secondary structures. And so in this case, you'd start with some start symbol S, and then it just has a bunch of grammar rules that convert into additional symbols until you're left with only dots and brackets. And then that generates a dot bracket sequence, which, as we discussed, is in bijection with these RNA secondary structures. So I want to look at one grammar in particular that's been used successfully in the literature and has been shown to have good accuracy, which is the Nuts and Hine 1999 grammar. And so it just has a bunch of rules here that start again with that S symbol, eventually get only parentheses and dots. And then each of these rules are associated with probabilities. And so the grammar has some parameters because you can choose those probabilities to maybe fit whatever scenario works best, although there are standard parameters that were shown to work very well for large classes of RNA. And so one thing about this is that it doesn't actually generate all possible Motzkin paths or dot bracket sequences, because this rule right here ends up forcing there to be a dot in the middle, which is actually a more biologically realistic thing anyway. Okay, so with that grammar then, I want to track like what is the distribution on the exterior loop for, again, the degree and the length of that exterior loop. And in order to do that, I'm going to need to add additional symbols into the grammar, S exterior and L exterior, that just track the transition between the exterior loop and the rest of the structure, but otherwise don't change the distribution of structures that this gives, which is not too bad to do. But once you've added those parameters, then you can just do this sort of standard Chomsky-Schutzenberger theorem to convert all of the grammar rules into a bivariate generating function. And then again, try and use the same process that we talked about. And so once I push through that pipeline, then I get, again, some results about degree and length. And it turns out that even in this more complicated context, the degree is still given by 1 plus a negative binomial. The parameter of the negative binomial is now more disgusting, and it just depends on the parameters of that stochastic context-free grammar. But the point is, with the sort of standard parameters, it still has a relatively low mean invariance. And then you could do the same thing for the length. And again, this looks more gross now, but it's still in the same form as the distribution was for the Motzkin paths. Okay, so we've sort of concluded that for all of these different models, the complexity of the exterior loop is maybe not that surprising. And so then a follow-up question would be, well, what aspect of the RNA secondary structures is surprising then? And I'll give you one option, which is we could consider how long are the helices that touch the exterior loop. And there are biologically meaningful reasons for why you might expect those helices to be longer than other helices, which is because when RNA structures are folding, they have multiple options for what to fold into. But if they have a really long closing loop, then they're probably going to find that closing loop relatively reliably, which will add some stability to the rest of the process. And so we could push through the analysis of the length of that helix or the exterior helices for any of the models that we talked about. And again, we find distributions actually geometric this time with really small means. And it is true that these means are way smaller than what happens in practice for the databases of RNA sequences. Although you could argue, which I think is a fair criticism, that maybe this is, right, these models weren't intended necessarily to be precise enough to talk about those lengths. And so for the same reason that I have to be careful about saying in 3D, it's surprising that the ends are separate. I don't want to do the same thing with these models. Okay, and then the last thing that I'll talk about is that there is a sort of direct connection between the sorts of parameters that we're talking about here and maybe parameters of pattern avoiding permutations. And so recent, well, work by Sergio Elzald and Mansour showed that you could define a type of Motzkin permutations that are corresponding to Motzkin paths, first by taking a bijection between Motzkin paths and Dick paths with no 3 consecutive upsteps, which is a relatively simple bijection, although I don't quite have time to go through those details. And then there's sort of the standard bijection between Dick paths and 3-1-2 avoiding permutations, where you just take the Dick path, you sort of rotate it until it is along this line right there, and then you put X's underneath the corners of it. And so here we have this permutation where it's saying 1 maps to 1, 2 maps to 3, and the X's just tell you what the entries of the permutation are. And so if I'm talking about close to, but not quite, the number of mountains in the corresponding Dick path here, then that really corresponds to a block decomposition of this permutation. I should have said too, and I skipped over, when I'm talking about these Motzkin permutations, they are then 3-1-2 avoiding and also don't have a consecutive 3-2-1, which is just from this no 3 consecutive upsteps condition. And so the results that we had already can tell you about the distribution of blocks in this block decomposition. It doesn't quite correspond to exactly the parameters I talked about, but it's close. And so it's asymptotically distributed again as 1 plus a negative binomial. It's just that the parameters change from 1/2 to 1/3. All right, that's all I have. Thank you.